Hi, and welcome to the 26th Ecological Continuity Trust webinar. Just some general housekeeping before I introduce our speaker. Please keep your videos off and your audio muted. If you have any questions, you're very welcome to put them in chat as you think of them, and Ben will chair a discussion and call on people to talk about their questions at the end. So I'm very pleased to welcome Raj Whitlock from Liverpool. Raj runs the Buxton Climate Change Impacts Laboratory, which is an amazing experiment. Having worked there myself on multiple occasions, I can testify to what a fantastic experiment it is. Raj took over from Phil Grime, who established the experiment and ran it for over 20 years. So Raj has been looking after the experiment for the last 6 years as part of a team with Emma Sayer from Lancaster and Carl Evans from Sheffield. So Raj, over to you.
That's great. Thanks, Carly. First of all, thanks to Ben and the ECT for their invitation to speak. It's a real honour to be able to talk to you about 30 years of research at the Buxton Climate Change Impacts Lab, but I'd like to also say how daunting that also is to try and put together a presentation that covers key highlights of the research at this excellent site over those 3 decades. The work, as you can understand, is going to be the work of very many people, not only the people who you can see named on this slide. I'd also like to point out that the experiment was set up by the late Phil Grime, who was my PhD supervisor when I was in Sheffield 23 years ago. Phil sadly passed away in 2021, and this site is very much a part of his legacy. So what I'm going to do is first introduce the site in a bit more detail, and then give you a flavour of some of the highlights of recent research at the site and some of the historical context of that as well. And I hope to give you a flavour of some of the outstanding research questions in grassland ecology, but in this site in particular, and a flavour of what could come in the future too. So the Buxton Climate Change Impacts Lab then is set up on an ancient sheep pasture on a dale side in Derbyshire. You can see an image of the site here. So the treatments have been set up since November 1993. The warming treatment started during November 1993 with drought treatments and summer watering treatments following in 1994. That means that the treatments have been run for 29 complete years, and that we're now in the 30th year of manipulation. The grassland at this site is a species-rich grassland over a renzinous soil. The soil is calcareous, varying in pH from around 7.5 to 6, and it has very high levels of plant species richness and genetic diversity. So typically in a grassland like this, we can find up to 40 vascular plant species rooted within a single metre squared. Perhaps one of the reasons why we're still going at Buxton, why the manipulations are still running, is that initially within the first 15 years, the vegetation was found to be resistant to climate change. And I'll say a little bit more about that later. The experiment is arranged as a series of 5 blocks spread out over the Daleside slope. I'll see if I can get the laser pointer working. Let's see. There we are. So here are the blocks, A to E, spread out over the slope. And within each of those blocks, we have 9 plots. Each plot of grassland is 3 by 3 meters. And it contains, as shown in the cartoon down here, a considerable variation in soil depth ranging from around 0 centimeters to greater than 40 centimeters. In each plot, because of the nature of these species-rich grasslands, you can get real populations of each species of plant present within just this small area of 3 by 3 meters squared. And this is part of the reason why we can have such high genetic variation within the plant species as well. So around 40 species, as I said, would be typical. And for some species, we've got many hundreds of individuals within each of those plots. How about the treatments? We've got 3 main treatments, and I'll introduce those to you briefly now. There is a summer drought treatment that's run between July and August each year. So it's due to start, this year's drought is due to start tomorrow. And this week, Andrew Askey's been busy setting up and that treatment ready to run. It's a series of automated rain shelters. And what happens is there is a sensor halfway up the slope there, which when rain is detected during July and August, it will drive these rain shelters up and over the plots. When it stops raining, those shelters will retreat to an off-plot position. We know that this has an impact on soil moisture, which we have measured, and that's what the graph on the right shows. There's time along the x-axis there and soil moisture on the y-axis. And what we see in a drought-treated plot is a reduction in soil moisture levels to around 5% in terms of the volumetric water content of the soil. So it's not the case that when it rains, surface water disturbs that. These are very freely draining soils, and we see a real effect of the drought treatment on that soil water. The warming treatment is implemented using a series of heating cables at the soil surface. And there we're doing winter warming between November and April, at the end of April each year. And there we are raising the soil surface temperature by 3 Celsius. This uses a series of small Linux computers, Raspberry Pi computers housed on the slope. And what they do is they interpret incoming information on soil surface temperature in the control plots and use that to switch on and off the heating cables in the warming plots to maintain an increment of precisely 3 Celsius. And you can see the way that works in the graph to the right there. So you can see how that warming temperature, that's the red curve there, very closely tracks the ambient temperature in the control plots, yet 3 Celsius above. The watering treatment is a summer watering treatment that's between June and September each year. And here we are adding 20% of water over and above the long-term average. And so the impact of that you can see in the photo there during the extreme drought of 2018, which I'll also talk to you more about later. The watering plots there are visible as the green squares, which have been rescued from the effects of that drought by the watering treatment. Importantly, since this is an experiment, we have control plots, non-manipulated plots, and we also have 2 combinatorial treatments, heated or winter warming plus drought, and warming plus watering. And I've got some icons there to describe those treatments. Okay, so this experiment has been running for a very long period of time, but it's not actually the world's longest running climate manipulation. That sort of accolade goes to a warming experiment in the Rocky Mountain Biological Lab that was set up by Professor John Hart at UC Berkeley in 1989. So this is a warming experiment over a meadow. And that experiment, so I've had some recent correspondence with John, was run for 30 complete years. So the manipulations there actually ceased in 2019. So another very, very long running experiment. But John cited one of the issues with experiments when they're run for this long is that the equipment that's being used can deteriorate over the time. So one of the problems at the Rocky Mountains Biological Lab was that the equipment, in John's words, had kind of taken a real beating. And although they would have liked to continue that experiment, they they were unable to find the funding to support that, and it would have required quite significant overhaul to infrastructure. So the manipulations there ceased in 2019, and they're now following up the impacts of the climate change on the vegetation to see how quickly the community recovers, or whether it does indeed return to its original state. So the question is, I guess, why would you continue such an experiment beyond 30 years, and why do we want to do that at Buxton? It's certainly not for the to win the accolade of having the world's longest running climate manipulation experiment. And I hope through this talk to convince you that there's good scientific reasons for continuing the manipulations. I guess one of the obvious reasons why it would be useful to continue manipulations at Buxton are that the impacts of climate change take place over many years. So this figure here shows the expected impacts of different ecological processes on vegetation and their approximate timing. And these are this is a guide, it's a sort of schematic diagram. So the x-axis isn't intended to be a sort of precise guide there. So at the top there, we have species expansion and relocation along local gradients. So that the organisms that are present in the ecosystem effectively become selected by the climate treatments or by the climate. And so the ones that are particularly well adapted can expand and others decline. And that process has been detected at Buxton, as you'll see, and is expected to sort of play out over decades. Eventually, what we expect as climate change proceeds is that there will be a decline in regeneration by those resident species, and eventually perhaps also local extinction. There's evidence, although we haven't lost any of the plant species from any of the treatments at Buxton, there's evidence that some of those species are at that point now where we may well lose them from the climate treatments, some of the climate treatments. Something I won't talk about is the immigration of new dominant species. So through time, we also expect perhaps species that are not present at the site but could be well adapted to the conditions there could immigrate and become established and perhaps become new dominant species. These might also include invasive species whose range is expanding as the climate changes. And the last process there is a phenotypic and genetic restructuring of the populations of each of the species present at the site, and that's expected to occur, up to the time frame of perhaps 50 years and beyond. And I will say a little bit about that. As you can see, there are a number of different important ecological processes here taking place over perhaps different time frames. And so the impact of climate change on the system will also depend on how these interact with each other. And that aspect of the complexity of the ecological response, I think, requires an experiment that goes on across or spans this time frame. And that's a good reason for continuing manipulations. There is also the inherent complexity of ecosystems and the fact that the impacts of environmental change, in this case climate change, can take place both directly and indirectly. And by that I mean that all individual organisms or species can experience the climate and respond directly to those changing conditions, but also indirectly via their interactions with other coexisting species. And those might be for plants, other plant species, or perhaps also soil microbial species. And so this complexity also, I think, requires an experiment that has longevity, because we need to be able to detect the effects of the interactions between the different species as they respond to climate change, and then perhaps use other experiments to dissect the mechanisms underpinning those. So the aims here are to give you 5 short stories that give you a whistle-stop tour of some of the research that we're doing. Also to illustrate some of the gaps in the knowledge and perhaps opportunities for the future. And I will be touching on the extent of climate-induced ecological change at this experiment, the evidence that we have resistance or resilience in the system and the strength of this, some of the mechanisms that might be underpinning it. I'll comment where appropriate on the strategies that underpin this. So where are species responding through stress tolerance? Where might they be responding through adaptation to resource exploitation? And I'll also talk a little bit about the coordination of responses between plants and soil and implications for grassland management. So first of all, story one, the plant community. So how we measure change in the plant community has changed over the years. In the first 15 years, up until 2008, we used point quadrat analysis to understand changes in the vegetation. Since 2008, we used a fixed quadrat approach, and the fixed quadrat is actually very small. It's shown here 10 by 10 centimeters. And the person who's been heavily involved in all of our surveys up until 2018 is Phil Grimes, shown here in 2018 in the last vascular plant survey he did at Buxton. What we do is we count the cover of each of the plant species in this 10 by 10 centimeter area. And we have these quadrats replicated right across the experiment. So we have 240 of these permanent quadrats in total. Within each treatment, those quadrats are spread over the soil depth gradient. So we know which quadrats are on deep or shallow soils, and we know the precise soil depth. We also know something about the soil conditions. So we know the pH that has been measured, and for some of these microsites, the levels of soil nutrients also. So that's the system within which we now collect vascular plant data. And so we've done some major surveys of the site since 2018. As I mentioned before, up until 15 years, we thought that there was resistance to the climate treatments. That was based on the fact that we had not lost any of the species from the plant communities, but also using the point quadrat method, there was very little evidence actually that the structure of the plant community itself was shifting. When we moved to the quadrat-based approach, one of the things that we picked up straight away, and you might imagine this given that we had good information on soil depth within these microsites, was that the climate treatments were driving local restructuring of the plant community, such that species could find niches within each of the treatments based on their affinities to the soil depth gradients. So, for example, species that were more able to tolerate resource deficit can be associated with these shallow soils, and those which are more competitive perhaps could be found on the deeper soils. Within a treatment like the drought treatment, that also means that there might be niches for drought intolerant species on those deeper soils because of the fact that moisture will be retained in the soil for longer there. Using the quadrat-based approach, we've also been able to show that there is a clear separation in the plant community structure of the different treatments. And here those treatments are shown in different colours with letters denoting the treatments. So HD is heated drought, D is drought, C is control. W is watering, H is heating. For each of those treatments, you can see these vectors here connecting 4 points, and they give a timeline through the experiment. So 2008 is the 2008 survey. Then we've got 2012, 2016, and 2019. So you can also see that through time, the structure of the plant community is not constant. It's drifting through time. But there is a clear impact of the climate treatments there on plant community structure. We also know from earlier work that the biomass productivity has been impacted by the climate treatment. So in particular, the drought treatments there are producing less dry biomass. The same pattern is evident for plant species richness as well, with reduced richness in those drought-treated plots. We also know that the change in plant composition is not random. Actually, we can explain part of the response to the treatments based on the traits that plant species have. So illustrated here on the x-axis we have leaf construction cost. That's the measures part of the leaf economic strategy of the different plant species. On the y-axis, the response of those plant species to moisture availability in the treatments. So above 0, we've got plants that do better under the watering treatments, less well under drought, and below 0 we have those plants that are being benefited by the drought treatment. Anyway, those responses to the climate treatments are partly explained by plant traits, raising the possibility that we can use traits to, at least in this experiment, to predict responses to the climate treatments. So here, what we can see in overview is that plants that are responding well to the drought treatment have greater leaf construction costs. So they're moving towards, perhaps we're moving towards here a strategy of stress tolerance in the drought treatment. So given these responses, one of the questions that we sort of opportunistically sought to address in 2018 was, do these responses to the climate treatments in the plant communities set up the vegetation to be more resistant to extreme events? For example, the shifts in the drought-treated communities might mean that they're better able to tolerate extreme natural droughts. Well, in 2018, we had such a drought that began in May and in the northwest of England lasted through to the end of July. In June 2018, as the map on the left there shows, there was very, very little rainfall, and the standardized precipitation index was very low. And in fact, in terms of drought and rainfall, the period May to July actually exceeded or beat the previous record of that was held in 1976. This was a particularly dry start to the year after a wet winter. And on freely draining calcareous soils, the impacts of such drought can be quite severe. And in the top picture there, as I've shown you before, you can see the impact of that visually on the vegetation. So the browning of the vegetation here in July 2018, it wouldn't normally look like that. And a close-up of one of the drought-treated plots there showing the extent of sort of above-ground senescence. So the question then was, are the drought-treated plots actually better able to withstand this? And so we carried out a survey in 2019 to see if we could answer that question. Why 2019 and not 2018? Well, we wanted to be able to discriminate between confirmed impacts of the drought on each of the plant species and the senescence that was observed in 2018. So although we observed above ground senescence of plant tissue, we couldn't be sure that that was would represented sort of death of plants or the reduction in their cover. So we surveyed in 2019, the year after that drought. And what we saw was a loss of plant species in all treatments except the heated water treatment. The water treatment is actually not shown on this plot here at the bottom because it's an internal control. We're using that to control for the year-to-year variation that you'd normally see. So all of these treatments you see here, control, drought, heated drought, heated water, are being controlled internally to the water which didn't experience this extreme drought. Anyway, it can be seen that the drought-treated plant community, that previous drought treatment didn't buffer that vegetation from further changes and loss of species. With the data we have, we can actually do better than that. So we can start to look at the dynamics a little bit. So we can start to decompose that change in species richness into the gains and losses of species. In any quadrat, as you measure over time, you'll find some resident species get lost, and others might be gained by recruitment via seed or perhaps clonal growth. And so we can separate these using the data that we have. And it turned out that the control and heated plots, they were experiencing a loss of species essentially through losses of existing species within those quadrats, whereas the drought-treated plots were better able to retain species that they already had. However, the drought-treated plots where they were losing their species was by an inability to gain species from outside the quadrat. So there was the sort of demographic buffering capacity, if you like, of those drought-treated communities had been impacted by the preceding treatments. And so they were unable to recharge the species that as gains from the outside of those quadrats. So there's kind of contrasted the treatments differed in the extent to which those processes explained species losses. And so here, I guess another question is, will those communities come back? This might suggest that the drought-treated plots have been pushed towards their limits in terms of their ability to resist those climate changes. Will those plant communities come back? Will the structure of those plant communities return? This is the position in 2019. Let's look at the control plots. 28 to 2012 are those points down here. The plant community by 2019 had taken this large excursion and shift. And in 2020, 1 year later, showed no signs of coming back in the opposite direction. The next major survey is in 2024, so we'll have to wait until next year to find out what is happening there. But at the minute, it looks as though we could, in fact, be approaching a tipping point with some of these climate treatments, where a combination of the treatments themselves and their responses to natural extreme events are pushing them perhaps into new structures or new community structures which cannot be recovered from. So short story 2, the soil microbial community. So here, returning to this diagram, we expect that that plant community above ground and the changes will affect the soil microbial community below through the quality and quantity of inputs from the vegetation. Those could be from leaf litter or from root exudates, but also there could be changes in species interactions through mutualisms and antagonisms within that soil community. And I'll say a little bit about how that soil community is responding, and later on a bit about how inputs might be shaping that. So what we know is that the impacts of the climate treatments on the plant community are actually altering the trait distribution of that plant community, and that's shown here by this co-inertia analysis, which seeks to link species responses to the treatments through to the traits that they actually hold. And what we can see here is that some of the traits are quite tightly associated with the climate treatments. For example, leaf construction costs, the CC masses, positively associated with the drought treatments. Plant canopy height is associated with the warming treatments. To paraphrase quite a complicated graph, the drought treatments are creating resource stress, and they're selecting for plant traits that are related to resource use efficiency and potentially therefore generating low quality litter for the microbial community. The warming treatments, on the other hand, are lengthening the growing season, leading to an opportunity for competition and traits related to rapid growth and therefore high quality litter. These this environment, both the climatic environment and the environment of litter inputs and extra inputs, are actually driving change in the microbial community. And here we see evidence from metabarcoding, 16S metabarcoding. Samples for this analysis were taken in November 2017. Just to note that that is outside of the drought treatment. So these results are legacy effects of the drought treatment. They would represent legacy effects of the watering treatment, but the samples were taken during the warming treatment. So first of all, the ordination you see there, that this represents phylogenetic distance between different permanent quadrats out in Buxton. So all of the 240 quadrats are shown here. It's quite a complicated diagram, but essentially, as with the plant community, we have an effect of the treatment. So for example, here we see the drought treated plots in gold, quite separate to the watering treatment in blue over here, the control treatment in black, and the heated watered in green. So those are the colours. We also have these deep and shallow soils represented in filled and open points, and you can see all of the shallow soils, or most of them, the vast majority over on the left-hand side with the drought-treated plots. So we have an impact on bacterial community structure here that's likely largely explained by the effects of the drought treatment. The treatments also impact on the bacterial community's richness and evenness, and there's significant impacts of soil depth. Around 16% of phylogenetic dissimilarity in bacterial communities is explained by the treatments, and a further almost 4% by soil depth. So in total, quite a large proportion of differences in bacterial community structure actually explained by treatment and depth. So we know that in the drought treatment, the bacterial biomass is reduced, and this is from molecular biomarker work. So it's these changes in community structure take place in a context where our overall biomass of that community is reduced during the drought treatment. There's also evidence that specific taxa within the bacterial community are responding, particularly this abundant group, this abundant phyla here, the actinobacteria. Taxa within there are both increasing in response to drought and relative abundance and decreasing, and also show relationships with warming. Some of these other phyla here show relationships, additional relationships with drought, but most of the effects we see in the bacterial community are being driven by the drought treatment. With the soil fungal community, the picture is not so clear, and here we have much less separation in the fungal community by treatments. So there's no significant impacts of the treatments on fungal richness or evenness, no significant impacts of soil depth there as well, and only low levels of community dissimilarity are explained by treatment and depth, although there are some significant effects. As with the bacterial community, there's a big impact of drought on estimated biomass of that community. And we do have a few taxa within that community that show associations, either positive or negative, with drought and warming. From earlier work using TRFLP, we know that the shifts in microbial community are associated with the plant traits. That's all I can say there, really, because I've got to move on and wrap up so that it's time for discussion. And also, we know that those changes in bacterial and fungal community structure have affected how the plants respond to hormone signals that are present in root exudates. And this was an interesting story done in the lab, where we recovered soil from the different treatments, drought control and watered, and exposed them to soil to hormones, and then measured the respiration response. And what we found was that the treatments actually altered those respiration responses. Here you need to compare the shape within each of these graphs. So you've got sort of humpback shape with hormone concentration in that first graph in the drought treatment, but this sort of ascending pattern to hormone concentration in the water treatment. That's evidence that those communities are responding differently to those to hormone addition to the soil. We observe this also for other important plant signals that involved in mediating plant stress responses and defense. So that might perhaps mean that those bacterial and fungal communities are able to listen in to the plant signaling and perhaps even anticipate impending changes in their environment. Plant and plastic and genetic responses, again, have to be brief because we need to wrap up. But in terms of plasticity, we can use the experiment to look at plant phenology. We see the normal response to warming in terms of floral timing, earlier advances in the start, peak, and end of flowering, increased duration of flowering. But what Buxton adds is actually what happens to those kinds of responses when we manipulate precipitation. So with precipitation, additional precipitation, we actually see the opposite, so delayed start, peak, and end timings for flowering. Therefore, what happens when you combine those treatments? Well, actually, what we see is that the advance in phenology, the expected warming, is overcome by manipulation of the precipitation environment. And that shows you the importance of some of these combinatorial treatments. And provide some interesting new information about how phenology is sort of moderated. The warming response to phenology is moderated by other climatic factors. In terms of evolution, we do this by taking plants out of the soil and measuring their phenotypes in common gardens, and then extracting DNA and looking at what's happening within the plant genomes. We found, for example, that the most abundant dominant species at the site, Festuca ovina, shows a signature of local adaptation where plants from the drought plots actually do best under simulated drought environments and vice versa for control plants. And we've measured many phenotypes and shown that there's a consistent, there's an evolutionary change in the plant phenotype there. Equally, within the genomes, we've been able to identify genome regions that are responding to selection by drought. And I can't go into that in too much detail. But most interestingly of all, we find that some of the genes that are drought responsive are not the sorts of genes you'd expect. For example, we find overrepresented in differentiated plant genes, these genes that are related to plant defense and biotic interactions. And so, you know, this is a suggestion perhaps that the selection imposed by drought might be indirect and might actually be flowing from other organisms with which the plants are interacting. And with that in mind, I've got one last bit of data to show you, really, and that's to do with the plant-soil interactions. So we wanted to follow up that plant evolution and the changes in bacterial fungal community structure and see whether those changes in those compartments altered the plant-soil interaction and ecosystem processes. So we went back to Buxton, collected some bacteria from the roots of Festuca ovina, and we isolated them. So you can see a phylogeny of those pseudomonads here, both from the drought treatment and the control. So these are isolates that originate from individual plants in the field at Buxton. Because we've got the same effectively for Festuca ovina, we could set up an experiment where we manipulated the root microbial environment of populations from drought and control. So the drought plants with drought microbes, control plants with control microbes, and then the other two combinations. We also did this with soil wash from the field, and we tracked the phenotypes of plants and ecosystem processes. And one really cool result that came out of that was that some of the ecosystem processes depend not only on plant adaptation and microbial adaptation, but by an interaction between them. So soil respiration, for example, is increased when microbes come from the drought plots But when you do that with a plant that's adapted to the drought treatment, then that the genetic adaptation of the plant suppresses that respiration response. So this might imply that we've got quite a tight coordination between plant adaptation above ground and microbial adaptation below ground. And we see similar effects with some other ecosystem processes in the same experiment, which I don't have time to go into. And the final point that I'm not going to labour is that we're now using the findings from Buxton to inform the development of common standards monitoring of grasslands in collaboration with Natural England. So using our understanding of how the plant species respond to the climate at Buxton, we can begin to perhaps refine methods for monitoring grasslands so that they're better able to pick up climate impacts. And here we're examining some of the usual aspects that are measured using common standards monitoring, leaf litter, bare ground, and indicator species, and adding some additional quick and easy measures that we think can help embed climate response information in those monitoring techniques. So in conclusion then, that ecological change at Buxton is present and it's ongoing. We don't know how far this will go, whether a tipping point has been reached, at least in the vegetation, and whether those changes are now irreversible. It's apparent that soil microbial communities are also responsive to the climate treatments. Functioning is effective, we know that, but mechanisms and proximate drivers are unclear. So linking these things together, we're only at the beginning of that process. There is clear evidence for evolutionary adaptive change within plant populations, not just Festuca, but other species as well. Haven't been able to show those data. The consequences for species interactions are still not understood, but perhaps it looks like the evolutionary buffering might not be insufficient to stop change above ground, especially when we've got extreme climatic events. And there's this interesting adaptation and coordination between plant change at the evolutionary level and that shifted microbial community below ground and impacts on ecosystem processes. So lots more to unpick there in terms of the mechanisms underpinning that. And as with many experiments in the ECT network, there are big opportunities, I think, for informing on the real world management of ecosystems. I'm sorry for overrunning, but I'll leave it there and hopefully there's still a few minutes for questions.